In this video, we will be modeling the enzyme-substrate complex, as well as what happens when an enzyme gets denatured. Here's a list of what you will need to gather. Before we get started in the modeling activity, let's just make sure we understand what an enzyme is, though I get that modeling it is really how you're probably hopefully going to understand even more. But the definition of an enzyme is it's a biological catalyst. And catalysts speed up chemical reactions, but a biological catalyst means it's inside a living organism. And the way that an enzyme speeds up a chemical reaction is by lowering the amount of activation energy needed to get the reaction started. So if you look at this graph here, in purple, I drew the big hump, that large amount of energy that would be needed to be overcome in order to go from reactant to product side without an enzyme being used. And that's pretty much going to be too much of a barrier for life to occur in terms of reactions to happen quickly enough. So we need enzymes in our body to speed up chemical reactions so they can be fast enough to keep us alive. So if you see the green line there, that's showing the amount of activation energy being lowered, thereby speeding up the chemical reaction. And so when you lower that amount of activation energy needed, then the reaction can proceed forward. So what I want you to do is I want you to take two pieces of construction paper, each of a different color, and place them on top of each other. Next, fold them in half, and then after that, fold them in half again. And then open up the papers and cut along the fold lines. Basically, we're trying to get 4 equal sized sections. The reality is you only need one section. I just like to conserve paper. So grab one set of those two pieces of paper that are stacked on each other and then draw a groove across the middle. You can make this groove look any way you want. It's basically like as if you're drawing two puzzle pieces, one on top and one on bottom, that would be connected to each other. Make some kind of groove shape like that. Then simply take your scissors and cut along the groove. Now as you pull those pieces of paper apart, because you just drew on the one sheet of paper, the one that was on top, in my case yellow, there should still be fragments of that marker still visible where you cut. So what I want you to do is make sure that one that you drew on is at the bottom. So if you see what I've done is I've arranged the two different colors to look like they're puzzle pieces sticking together, but I ensured that the one that had the marking was on the bottom. So I had yellow. I have two sets, but I have yellow on the bottom in both situations, and you can see the markings on both of those. If you can't see the markings really well, I actually do want you to take your marker and make sure it's a defined groove on both of those bottom, whatever color you have, but in my case, yellow pieces. Then along that groove, label it on both of them with the term active site. So active site is S-I-T-E, not site like your vision, because it's a location. The active site is the location where the reactant of the chemical reaction that's going to be catalyzed by the enzyme will fit into the enzyme. And since we're saying that the active site is on the enzyme, I next want you to label those two bottom pieces in capital letters enzyme, just so it's real obvious and you remember that's representing the enzyme in this exercise. Next, I want you to label the top sheet of paper on both sides the substrate, and that is the reactant or reactants that are being catalyzed in this enzyme catalyzed reaction. So if you want to go ahead and place your top structure, the substrate, into that groove, the active site, to just visualize a substrate, a reactant of a reaction fitting into an enzyme which will speed up the chemical reaction. Go ahead and connect them, both of them. And what we've just modeled are the enzyme-substrate complexes that form. However, put one of them aside and just focus on one setup. So thinking about one enzyme with one substrate. Now keep in mind a chemical equation might include more than one reactant. So in our example, I just have the one piece at the top, so there's just one reactant, but it doesn't have to be that way. But we're going to pretend that this one reactant is going to be catalyzed, like the reaction is going to be catalyzed to turn into two products. It's going to break that reactant down. So I want you to now take your scissors and actually pull out the substrate, cut it right down the middle to visualize that the substrate has turned into the the products because the enzyme has catalyzed it. Now notice the enzyme itself did not change. It is not part of the reactant, it is not part of the products. It's simply something that can be used over and over again to catalyze different chemical reactions. Well, I shouldn't say different. Each enzyme is specific to one particular chemical reaction. And just to make sure you truly understand that reactants get transformed, turning into the products in a chemical reaction, I want you to cross off the word substrate that's on either half and write on each of the new pieces product. So we're visualizing a situation where one reactant, the substrate, was turned into two different products. Now let's put this into the context of a real chemical reaction. Let's pretend our substrate or reactant is hydrogen peroxide. There is an enzyme known to break down hydrogen peroxide, and it's called catalase. So your body happens to make hydrogen peroxide as a byproduct of some chemical reactions, but then your body needs to actually break that down. You don't want a buildup of hydrogen peroxide. So catalase, not to be confused with catalyst, anything that ends in ase tends to be a name of an enzyme, so if you see that suffix. And so this one is confusing because it sounds like catalyst, but it's actually catalase. And catalase is the enzyme that breaks down hydrogen peroxide. And what does it break it down into? It breaks it down into two products, water and oxygen gas. So what we just visualized then, if we kind of connect the dots, is when that substrate fit into the enzyme, and then when we, that was the enzyme like catalase with hydrogen peroxide fitting into it, when we then cut it down the middle, the substrate, and broke it into the products, turned them into the products, transformed them into water and oxygen, that was simulating that chemical reaction occurring. But now let's take that other setup, the one we didn't use yet. What I want you to do in this situation is I want you to imagine that something has impacted the shape of the enzyme. So I want you to pick up that bottom sheet of paper, in my case, the yellow one, but whatever one is labeled enzyme, and I want you literally to physically crumple it up. So why in the world would we do that? Well, sometimes there's something that happens to the structure of an enzyme rendering it useless. It's called denaturation. If an enzyme gets denatured, if its structure gets altered, because structure relates to function, if the structure is not intact, the function is lost, and it's lost permanently. So let's pretend that catalase was in maybe too hot of an environment, or maybe too salty of an environment, or maybe too acidic of an environment. If that occurs, then the catalase would get completely destroyed because its shape got destroyed, so the substrate can no longer obviously fit into the active site or be catalyzed in the chemical reaction, so it can't produce the products. So in this case, the hydrogen peroxide would just stay there. It wouldn't turn into the products because it couldn't, because the enzyme wouldn't be there to lower the activation energy to allow it to occur. Well, that concludes our modeling of an enzyme-substrate complex and also demonstrating how an enzyme can be denatured. Hope this helps. Construction paper biology. Like, share, subscribe, won't you please? Thanks!